Hello, Bio 250. My name's Sarah Martin, and I'm here with your news at 9. Tonight's top story is about a protein, specifically the melanocortin 1 receptor. But first, the weather. Take it away, Bob. Thanks, Sarah. In typical Rochester fashion, we'll be seeing sun, snow, and rain. Rochester really doesn't know if it wants to be spring or winter yet, but I'll let you know as soon as I find out. For now, I'm going to focus on the sun and talk to you a little about what the sun can do to you. The sun emits UV radiation. Even though we love to tan outside and soak in the sun, too much sun can be a bad thing and lead to sunburns and skin cancer. UV light exposure can damage our cells and the DNA within them. A photon of UV light can hit our cell's DNA, making the DNA increase its reactivity. The DNA can then react directly or indirectly. The reactive DNA can directly fuse two pyrimidine base pairs together or react indirectly through melanin. Melanin is a light-absorbing compound that, once hit with light, enters an excited state. Melanin typically releases that excitation as heat, which protects DNA from being reacted on. However, the system isn't perfect, and sometimes the energy cannot be dissipated as heat. This reactive melanin instead can bump into one molecule and then oxygen to create a reactive oxygen species that can ruin your DNA and damage it. So stay safe, class, and wear that sunscreen. Back to you, Sarah. Now, with more on melanin and the protein behind it all, Claire, our chief staff scientist, will bring us our top story tonight. Thanks for having me, and for that introduction to the dangers of the sun, Bob. Today, I'm going to be discussing an important protein related to melanin, the melanocortin 1 receptor. This protein receptor can be a major determinant of skin cancer, relative sun sensitivity, and skin and hair pigmentation regulation. The MC1R protein is the smallest member within the melanocortin family. This group of receptors consists of 5 members, MC1R through MC5R. The receptors vary in their respective tissue expression and functions, but are all G protein coupled receptors. MC1R is found in both melanocytes and leukocytes. Melanocytes are specialized cells that produce the melanin pigment that Bob touched on. Melanin gives color to different parts of your body, such as the hair, skin, and eyes, and melanin production stimulates UV resistance. A loss of function in the MC1R protein can create a fair-skinned, UV-sensitive, and skin cancer-prone individual. I'd like to first discuss the protein structure so that you can better visualize how it functions. The human MC1R protein is composed of 317 amino acids. The structure is made of 7 alpha helical transmembrane domains, which can be seen in this image. The transmembrane domain spans the cell membrane 7 different times. The alpha helical structures are shown in blue. The receptor is a G protein coupled receptor and has an extracellular N-terminus and intracellular C-terminus. This PDB structure shows 3 MC1R receptor complexes bound to the hormone alpha-MSH, a drug, afamelanotide, and a synthetic agonist called SHU-9119, which are shown in orange. The drug afamelanotide is used to fight a condition called EPP that is caused by the skin's reaction to sunlight. Agonists are compounds within our body that bind and activate receptors. This PDB structure used the agonist, drug, and hormone to reveal an interesting structural aspect of the MC1R receptor, the orthosteric binding pocket. This pocket is the receptor area where the agonist ligand binds. Typical agonists that interact with this pocket are melanocortin, the agouti signaling protein, and beta-defensin 3. An orthosteric binding site is composed of many amino acids that can interact with the agonists. Glutamic acid 94, aspartic acid 117 and 121, tryptophan 254, phenylalanine 257, and histidine 260 were found to be involved in the binding process at the orthosteric pocket. These amino acids are shown in ball and stick form here in this video. Here you can see a representation of the amino acid interaction that occurs when a ligand, depicted by the green sphere, binds the receptor. MC1R signaling is an important part of this protein's function. The protein is hormonally controlled by the previously discussed agonists that interact with the orthosteric binding pocket. Once the agonist ligands interact in the pocket, the G-alpha protein can dissociate from the MC1R protein, which creates adenyl cyclase activity. This cleaves an ATP to generate the second messenger molecule, cyclic AMP. MC1R signaling is carried out via this adenyl cyclase activation. Cyclic AMP stands for cyclic adenosine monophosphate, and it is a second messenger in signal transduction for a number of pathways, including energy metabolism, cell division, and ion transport. The activation of cyclic AMP signals for an upregulation of melanin production and its deposition into the layer of the skin known as the epidermis. Melanin helps function to limit UV light penetration into the skin and improves the nucleotide excision repair pathway that is used to repair the photolesion damage that the UV light creates in human DNA. This protection pathway prevents further UV damage by releasing more melanin and depends on the signaling of the MC1R protein, which helps alleviate skin cancer risk. The MC1R protein is involved in other melanin-based human processes, including hair and skin coloring. MC1R mutations are found in many red-haired individuals. Melanocytes make two different forms of melanin, eumelanin and pheomelanin. Different amounts of these different melanin forms are responsible for the various hair colors you see on people. Eumelanin helps protect the skin from UV damage. People with increased amounts of eumelanin typically have brown hair and dark tannable skin. People with pheomelanin usually are blonde or redheads and have fair skin that is easily damaged by the sun. The MC1R protein controls which type of melanin is produced by the melanocytes. This receptor uses the signaling techniques we talked about to create a signal transduction pathway to stimulate the production of eumelanin. If the MC1R receptor is damaged, mutated, or inhibited, the melanocytes make pheomelanin instead of eumelanin, which creates skin prone to UV damage and skin cancer. This is why the MC1R receptor has been investigated as a possible drug target to fight against skin cancers such as melanoma, basal cell, and squamous cell cancer. We had the chance to interview a local Syracuse resident, Addison Martin, who also is impacted by a melanin deficiency condition called poliosis. People can be born or develop poliosis and observe a white streak in their hair due to an absence of melanin and melanocytes. Poliosis can serve as an indicator of early melanoma due to it showing an issue with melanin production, which shows its relevance to the MC1R protein. Thankfully, Addison doesn't experience the more problematic symptoms that can be associated with poliosis, and instead just has a unique hairstyle.
My mom's jealous because she has to pay for her highlights at the hair salon. I got mine for free.
Back to you, Sarah. Thanks for the information, Claire. I didn't even realize how such small little proteins can play such a huge role in our day-to-day lives. I'll be sure to wear my sunscreen whenever Rochester decides it's time for summer. This is Bio 250 News. Thanks for watching, and we'll be back at 11.